Okay, folks, let's get started. I think it's the only time I've ever put the grades on the board and then the class didn't get quiet when I did it. So maybe that's good news. I don't know. The average on the exam was 73, as you probably saw on the online thing. That was up slightly from before. And overall, I was fairly pleased with performance. Of course, there's people who can improve and so forth. But for the class as a whole, I thought it was a pretty good job on the exam. So I was not unhappy with what I saw. The exam had one error on it, and that is there were 2 more points than I had intended in section 2. So instead of being worth 50 points, it was actually worth 52. So some of you may wonder, how did you have points off and still have 50? There were a couple who had that, actually. And that was why. So the total exam, if you got everything, including the extra credit, would have been 104 points. So for what that's worth, that's where it was. But I was, as I said, fairly pleased with that. If you have questions, as always, please let me know. I'll be happy to hear from you and talk to you about those and consider what you have in terms of requests for regrades and so forth. Okay. Last time I got a pretty good start on transcription, and today I want to get through as far as I can through that. The most interesting protein, I think, in transcription is actually Rho. And I talked about how Rho was this protein that literally climbed the RNA rope that was hanging off of the DNA open complex as transcription is occurring. And as I described, there is a sort of a race that occurs between the movement of Rho and the movement of the RNA polymerase. Once Rho catches up with the RNA polymerase, it destabilizes the polymerase, and what it literally does is, as I said, lifts the butt end of the polymerase up off of the DNA, and everything falls apart. Everything falling apart stops transcription. The way that this happens at a relatively specific, you can imagine it doesn't always happen at exactly the same nucleotide, but in a fairly specific range of the DNA, the reason that this termination is fairly specific is that RNA polymerase goes in what I call fits and starts. Okay? It goes faster through some sections and slower through others. In general, the more GC-rich the sequences are, the slower the polymerase moves. And that's not totally surprising given the fact that GC base pairs hold to each other tighter than do AT base pairs. So Rho helps facilitate that process. Now, Rho is an example of what I call a factor-dependent termination. Rho is a factor, and so this mechanism of terminating transcription depends on this factor. There's another interesting mechanism that bacterial cells use that facilitates the termination of transcription, and it's called factor-independent method. Okay? And it was actually that one that I had on the screen first last time. Factor-independent does not use a protein. Okay? So a protein does not cause transcription to terminate. Instead, built into the sequence of certain genes are sequences kind of like we see here. So this is an RNA that has been made. So the 5' end would be way out here. We made a long RNA. And near the 3' end, we have a sequence that looks like this. Now, as you can see, that sequence has within it regions that can form base pairs with each other. GC, CG, CG, blah, 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 all the way up there. Okay? Well, because they can, they do. They're close enough to each other that they will actually fold up into a double helix like you see there and form what we call a hairpin. This looks kind of like a hairpin. There's the head, there's the pin down here. This hairpin actually is a structure, okay? Just like a DNA double strand is a structure. And that structure is actually capable of bumping into the bottom end of the polymerase, because the polymerase is sitting right here. The polymerase is reading the DNA. The polymerase is sitting right here. Its butt end is right here where this thing is. Well, guess what? Lifting the butt end up made a difference in the Rho factor termination. So too does lifting the butt end up with the formation of this duplex also lift the RNA polymerase off of the DNA. And as a consequence of that, look at the last bases that the RNA polymerase was putting in. U's. UA base pairs fall apart very easily. So again, this sequence and this helix that we see here facilitates the termination of transcription without a protein factor. Okay? So very cool mechanism, but in general, the same sort of mechanical considerations, lifting apart of the polymerase off of the DNA facilitates everything falling apart. And when everything falls apart, of course, transcription is done. Yes, sir?
So why do the UA base pairs have to come apart for the polymerase to fall off?
Okay, why do these guys have to come apart for the polymerase to fall apart? That doesn't necessarily, but again, this is when we think about what's stabilizing a double helix, okay, then this is part of what's holding everything together. This RNA has got to fall off as well. This just simply facilitates everything falling apart. Yeah. It's a cool system. Okay. So that's transcriptional termination in prokaryotes. All right. Now, I'm going to spend just a couple minutes talking, or a few minutes talking about processing of RNAs in prokaryotes. And my reason for doing it is to, obviously, first of all, tell you a little bit about how some of these RNAs are made after they've been synthesized. That is, there has to be things that happen to them after the RNA polymerase finishes copying the DNA. But also it provides an interesting comparison to what we'll see in eukaryotic cells. Eukaryotic cells go to a lot more elaborate systems of modification of RNA after the RNA has been transcribed. Okay? So what we're talking about here is post-transcriptional modification, things that happen after the RNA has been transcribed. Okay? All right. Well, a prime example are those of the tRNAs and the ribosomal RNAs inside of prokaryotes. As this figure shows, they're actually made on one long RNA molecule. That long RNA molecule includes the 16S ribosomal RNA, which I'll say a little bit more about later in the term, the 23S ribosomal RNA, okay, and the various tRNAs that the cell has to make. Okay? Now, these are made in one long transcript, and of course, in the end, they're not in that one long transcript. They're actually contained in individual subunits. That means that each of these individual sequences have to be excised or have to be removed from all the surrounding sequences. So that's one post-transcriptional event that has to happen in the processing of RNAs in prokaryotic cells. There are ribonucleases that are involved in this processing. You see one called ribonuclease P, which generates the 5' end of tRNAs. What does that mean? It means that when I go back to look at this sequence, I've got about 60 tRNAs in here. Each one has a 5' end, so the ribonuclease P is involved in cutting to generate a 5' end, then it cuts the next one at the 5' end, cuts the next one at the 5' end, etc. That's what RNase P does. RNase P turns out to be an interesting enzyme. I won't say too much about it other than to point out that it's not a protein. At least its catalytic site is not a protein. And I'll say something more about that later. Ribonuclease 3 is an example. By the way, ribonuclease frequently people say is RNase. So I will say, in fact, I will usually say RNase instead of ribonuclease just because it's faster to say. RNase 3 excises the ribosomal RNAs. These are 3 different ribosomal RNAs that are in that transcript that you saw before. And again, this is a nuclease. It's an endonuclease, meaning that it cuts phosphodiester bonds internal. And ribonuclease P is also an endonuclease. It's cutting internal phosphodiester bonds. Well, as a result of action of those 2 enzymes, there are the bacterial cell then has functional ribosomal RNAs and functional transfer RNAs. Okay? Now, transfer RNAs in prokaryotes are the most chemically modified RNAs. Okay? Transfer RNAs in both prokaryotes and eukaryotes get chemically modified. We'll see that modifications to RNAs, all RNAs in eukaryotes, are much more extensive. But even in prokaryotes, the transfer RNAs are chemically modified. We don't fully understand the significance of all the modifications. Some of the modifications, for example, result in the creation of T's in the transfer RNA. Why? Well, it's not completely clear. Okay? There are other modified bases, and in fact, I'll show you a schematic in just a second that actually does that. The last thing that happens to tRNAs is at the 3' end of all tRNAs, there's the addition of a 3-nucleotide sequence, CCA. So the far 3' end of transfer RNAs have that sequence added onto the end of them, and it's actually onto the A that the amino acid will ultimately be attached. Okay, just as some examples of chemical modification, these are common chemical modifications that occur in transfer RNAs. You don't need to worry about the structure, even the names for that matter. I just want you to be aware of the fact that these chemical modifications do occur in transfer RNA. Here's the synthesis of ribothiamidylate. Notice how they didn't want to put the D in front of thymidylate for DNA, but they want to put the R in front of it for RNA. I say let's put it in front of both. But anyway, there's how we get T in transfer RNA. Pseudouracilate is something, as its name implies, similar to uracilate, but not identical. Okay? What's the difference? There's an N, there's not an N, so we see basically the swap of these guys here. Okay, let's see, what else does I want to say there? That's basically what I want to say about prokaryotes. I want to spend some time talking about eukaryotic RNA synthesis in general terms, and I'll spend another lecture or two later in the term, which means in about a week or two, talking about some details of eukaryotic RNA synthesis. So today's stuff will be fairly general, although you're going to see there's a lot of detail in it. Okay, well, let's first of all start out thinking about the differences between the phenomena of transcription and translation in prokaryotes versus eukaryotes. The biggest difference that we see is that eukaryotes have a nucleus, and the nucleus provides a barrier to translation. Translation is how we're making protein. That's the process of making protein from the sequence information in the messenger RNAs. In prokaryotes, there's no nucleus. So when a messenger RNA starts being synthesized and the tail starts hanging off, ribosomes, which do the translation, can start to bind to that messenger RNA sequence and start translating at the same time as transcription is going on. And the first question you're thinking about was, how does Rho get in there and do its thing? It does. That's as much as I can tell you. Rho is able to navigate all this stuff and terminate transcription as it needs to. But transcription and translation are going on essentially simultaneously in prokaryotic cells. That can't happen in eukaryotic cells, because in eukaryotic cells, the transcriptional machinery is located in the nucleus, but the translational machinery, that is the ribosomes, is located in the cytoplasm. So in eukaryotic cells, transcription and translation cannot occur at the same time. And that turns out to be interesting and important because, as we're going to see, RNAs in the nucleus are significantly modified in eukaryotic cells. This allows all of that modification to occur before the messenger RNA makes it out to be translated. So a lot of modification happening to the RNAs in the nucleus of eukaryotic cells. Okay. As I mentioned in the lecture on Monday, there are three primary RNA polymerases in eukaryotic cells: RNA polymerase I, RNA polymerase II, and RNA polymerase III. And these RNA polymerases have specialized functions, and the specialized functions relate to the classes of the RNAs that are being made. RNA polymerase I works on the larger ribosomal RNA molecules. Okay? The larger one. There's one small one that is not made by RNA polymerase I, but all the larger ones are made by RNA polymerase I. Okay? You can think of all this as nucleus. Nucleolus is just a specialized region of the nucleus. Okay? So we're not going to distinguish different regions of the nucleus. RNA polymerase II in eukaryotes is the one that is responsible for making messenger RNAs, okay, and small nuclear RNAs. And I won't say really anything more about those here, but for our purposes, we're interested in the messenger RNAs that RNA polymerase II makes. RNA polymerase III is involved in making a lot of small RNAs. These include the transfer RNAs as well as the smallest ribosomal RNA. Now, you'll notice the third column here. The third column says sensitivity to alpha-amanitin. Alpha-amanitin is a deadly poison. It's actually the deadly poison that kills people in death cap mushrooms. Okay? If you eat death caps, they call them death caps for a reason. All right? What happens is death caps have this compound called alpha-amanitin. I'll show you the structure in a second. And alpha-amanitin will completely nail your RNA polymerase II. If you can't make messenger RNA, you're toast. The most common thing that happens when people eat death caps is they get sick fairly quickly. And the reason that they're getting sick is because any sort of unusual compound gets transported to your liver. This alpha-amanitin makes it to the liver and basically starts killing the liver quickly. Your liver function you can't live without for very long, and you will get sick. Okay? Alpha-amanitin poisoning from death cap mushrooms is fairly rapid. If you should consume death cap mushrooms, you've got literally a few hours to get a liver transplant. That's where you are. Okay? Now that is nasty. Okay? Because as you know, getting organs donated is not easy. And getting it done within a few hours is not easy as well. Questions?
Is that the Amanitin hysteria? Is that what you mean by death cap?
Mm-hmm. Yep. Yep. Yes?
Are there cases where it will be quick enough to get a liver transplant?
Yes. There are people who are alive because they were able to get a liver transplant and survive that. The numbers are low. Okay? And there are people every year who go out and eat mushrooms. They don't know what they are, and discover this unfortunate fact very quickly. Yeah. Other questions? Okay. So, very nasty compound. As you can see, it affects all three. Well, it says insensitive. It actually takes fairly high amounts to inhibit this guy. But all three actually are inhibited to some extent by alpha-amanitin. So it's a very deadly compound. There's the structure of it, and probably the worst part of it is memorizing the structure of it, if you were to do that, right? I mean, I wouldn't want to do that. And the source of the material is this guy right here. There's the death cap mushroom. He says, oh, I ate one of those the other day. I'm okay, right? So I'm resistant to death caps. Ha, ha, ha. Okay. You're not resistant to death caps. In fact, the sensitivity to alpha-amanitin is not just people. Most eukaryotic cells have their RNA polymerase strongly inhibited by alpha-amanitin. It's actually used as a research tool to study processes in the absence of transcription in the laboratory. Okay, well, we have 3 different RNA polymerases, and we, as a consequence, have 3 different types of controls of the 3 classes of genes. Okay? Now, I'm not going to go through all the details here. My main reason for showing you this is to, first of all, point out that there's a lot more complexity to the controls of transcription in eukaryotic cells than there were in prokaryotic cells. This only begins to scratch the surface of the complexity, especially for RNA polymerase II encoded genes. Okay? The number of things in here called elements that can affect RNA polymerase II is significant. Now, another thing that we see in control of transcription in eukaryotes is the fact that the RNA polymerase itself isn't the thing that binds to the promoter. It's usually helped by a mix of other proteins called transcription factors. So we talked about transcription factors a little bit on the last exam. Or did we? I guess we didn't, did we? Yeah, I'm teaching. See, I'm teaching 3 on eCampus classes at the same time, so I always lose track of what I've said to who. So my other class, we've talked about that. So there are transcription factors that are important for controlling whether or not transcription occurs. These are proteins that bind to DNA. So proteins that bind to DNA are called transcription factors if they affect transcription, obviously. Okay. Well, what I want to do is say a little bit. I'm not going to say really anything about polymerase I promoters or polymerase III promoters, but I do want to say a little bit about polymerase II promoters. These are the control regions ahead of the messenger RNA sequences. Okay? Well, no surprise, we see a conserved sequence ahead of messenger RNAs in eukaryotic cells like we saw in prokaryotic cells. There's a TATAAT box again, TATAAT. It's not the same as it was before, but it's very similar to what we saw in bacteria. Okay? The numbers out front are the percent of the time you see each of these bases in that appropriate position. The TATAAT box in eukaryotic cells is further away from the start site than it is in prokaryotic cells. In the prokaryotic cells, we call it a -10 sequence. In eukaryotic cells, we tend to find TATAAT boxes between about 30 and 100 nucleotides ahead of the start site. That means it's quite variable in the exact positioning. We see a lot of variability in positioning in eukaryotes, a lot more than we saw in prokaryotes. I'll say, again, a little bit more about that later. Okay? Not all eukaryotic genes have a TATAAT box. Not all eukaryotic genes have a TATAAT box. Those that are made at high levels, that is, that are made a lot of, will have a TATAAT box. But there are examples of eukaryotic genes that do not have a TATAAT box. Other common sequences that we see ahead of the transcriptional start site in eukaryotes include something called cat boxes. And these are really smelly things. Okay? They have a sequence that looks something like this. And another common one is called a GC box, and it looks like this. Now, you would probably guess this one isn't involved in strands being peeled apart, and you'd be correct. It's just used as a recognition binding site for a protein. These sequences aren't found in front of all genes, but they are not uncommonly found in front of many genes. And their positioning is also variable, usually between about 40 and 150 base pairs of the start site. Now, 150 base pairs of the start site, or 100 base pairs of the start site, is a little harder for us to envision than we had for the prokaryotic cells. When we talked about the prokaryotic cells, I showed you the picture, the RNA polymerase sitting down on that promoter and positioning itself at the start site where it could move forwards. And it covered about 30 to 40 nucleotides. So one question you might have is, well, is the RNA polymerase in eukaryotic cells so big that it'll stretch 150 nucleotides? And the answer is it's not. Okay? The complex that I will be describing will stretch out some, but even there, it won't necessarily stretch for a full 150 nucleotides. Okay? Well, how do these things extend their reach, as it were, from the place where they're binding to the place where the transcriptional start site is? The answer to this is that DNA bends. Okay? DNA coils around, and we're going to see some real big examples of that when I talk about another group of control sequences called enhancers. Enhancers may be located up to several thousand bases away from the transcriptional start site. Okay? So that means that DNA has to be able to coil around. So if I bend something around, I could take a sequence that is 5,000 base pairs away from the start, bend it up here, so that now they could be in contact and help facilitate the initiation of transcription. And that's exactly what happens. Okay. So these sequences, of course, are bound by some proteins that help to facilitate the start of transcription. When we talk about the start of transcription, actually, before I do that, let me jump around for a second. I want to talk about the last sequences, which are called enhancers. So that's this region right here. I don't have a good figure to show you. But enhancer sequences are really interesting. When they were first discovered, people were very, very puzzled by them. Okay? What they discovered was that they found little sequence stretches, kind of like we saw here, cat boxes, GC boxes, or TATAAT boxes. And they found that those sequences, those short sequences that they found, the sequence varies, so there's no consensus. It depends on the individual enhancer. Those enhancer elements could be located up to several thousand base pairs away from the transcriptional start site. That could mean it was ahead of the transcriptional start site. In some cases, it was in the middle of the gene that was being transcribed. In other cases, it was located way 3' or downstream of the gene. There were no known control sequences that behaved like enhancers did. They were very puzzling, and people studied them pretty intensely and they knew that they were functioning, because if they took the sequences out of the DNA was no longer transcribed in the same way. Okay? Well, enhancers are bound by enhancer-specific proteins. And each sequence has a specific protein that will bind to it. We don't see enhancers in prokaryotic cells. Your book makes some hand-waving gestures about prokaryotic cells, but really we don't see enhancers in prokaryotic cells. We only see them in eukaryotic cells. And it appears in eukaryotic cells that the enhancers play important roles in controlling tissue-specific transcription. Very important roles in controlling tissue-specific transcription. Okay? I'll give you an example. Let's think about our muscle cells. Our muscle cells need to make certain proteins that are involved in muscular contraction. Okay? My skin cells don't need those same proteins. So what I would like to have is a mechanism that will tell the muscle cell, make this protein, but that same mechanism doesn't work in skin cells, for example. Make sense? There are thousands of examples, but there's one example for you. Well, how do cells control that? A very simple way to control it is if the muscle cells make a protein that binds to the enhancer for those genes, and the skin cell doesn't make that protein. It's a very simple control. Muscle cells will make the enhancer protein will bind to the sequence, turn on the transcription of that gene. It will work in muscle cells. The same protein will not be made in skin cells. Skin cells don't make the protein. They don't waste the energy on it, etc. The next question is, well, how does the cell determine which one of those to turn on and turn off? What we see is there's a hierarchy of controls. And those hierarchy of controls are built into the phenomenon of development. Okay? That's beyond the scope of this course. But those enhancers are very, very important in allowing that specific transcription to occur in certain cells and not others. Okay. Let us consider the phenomenon of initiating transcription in eukaryotic cells. Okay? Transcription in eukaryotic cells is a very orchestrated process. It's not the simple process that occurs in prokaryotic cells. Okay? In eukaryotic cells, there are a series of proteins called transcription factors, as I've already used that term, that bind to control regions near the transcriptional start site. So the things that I am describing here are not enhancers or enhancer proteins, but rather proteins that are recognizing those control regions close to the transcriptional start site. These transcription factors are named according to the RNA polymerase they work for, and they have letters designating different proteins. So, for example, the first protein to bind to a promoter region for most RNA polymerase II transcribed genes is a transcription factor labeled D, as in David. Okay? We call that transcription factor TF2D. standing for transcription factor, 2 standing for RNA polymerase 2, and D standing for the specific protein. Transcription factor 2D is a multi-subunit protein, and one of the subunits actually recognizes and binds to the TATA box. And that's the thing here called TBP, TATA binding protein. Okay? Now, of course, if you don't have a TATA box there, then that protein may not play the same role in initiating transcription. Again, that's getting a little beyond where we're at here. All right, then other protein factors come in and make the transcriptional initiation complex. Notice we don't even have RNA polymerase there yet. We see binding of a protein factor called A, that's TF2A, followed by TF2B. And do I care if you memorize the order of these? No, I don't. I do think you should know TF2D is the first one, and I think you should know that TBP is a component of that. There are at least 3 other proteins. And by the way, this is a general scheme. There are variations in this for other promoters that are used by RNA polymerase II, but this is a fairly general, good scheme explaining how this process occurs. Other proteins come in, and in fact, they're actually shown here in the order that they come in. So you see D followed by A, B, F, E, H, like so. H acts kind of like, it actually has a dual function. H acts kind of like a helicase in that it helps to pull strands apart. It's not pulling them apart at 6,000 RPM like it is in the DNA replication scheme. And H also plays a role in phosphorylating the end of the RNA polymerase. Phosphorylation of the end of the RNA polymerase occurs at the carboxy terminus sequence. It's called the CTD, the carboxy terminal domain. That's the CTD right here. This carboxy terminal domain phosphorylation is necessary to enter the elongation phase where the RNA chain is being made. So we can think of the phosphorylation as a transition between the initiation phase, when all of the binding is occurring, and the elongation phase, where the phosphodiester bonds are being made. Okay? It's slightly different than the prokaryotes. In the prokaryotes, we have the initiation up to the first 10 nucleotides. That was the initiation phase. Here, we're calling the elongation phase after we've started putting things in. Okay, well, once this phosphorylation occurs, most of these factors fall back off, and polymerization occurs with the RNA polymerase moving to the right. As we can imagine here, we see the RNA coming off. Okay. TATA binding protein, and I guess that tells you that TATA binding protein binds to TATA boxes. TATA box binding protein makes a complex, and see that you can see it right there. And, okay. I talked about transcription factors. So some of those things that you saw there are transcription factors. Some transcription factors are specific for certain genes. Okay? Here is an illustration of the control region ahead of a gene involved in heat shock in eukaryotic cells. And yes, both eukaryotic and prokaryotic cells make heat shock genes. Okay? What you see on the screen is not at all uncommon. You see this region up here, heat shock element. Wherever you see the word element used in conjunction with a DNA sequence, they're talking about a specific DNA sequence. In this case, this specific sequence is bound by control proteins for turning on the heat shock genes. Now, you see multiple of them. You didn't see things like that in prokaryotic cells. Multiple elements actually allow the cell to have some fine level of control. Let's imagine that if we measure the strength of this promoter by how many elements bind, the cell is being heat shocked. It might be making a lot more of these control proteins and really turn this on. If we have very little heat shock going on, only one or so of these elements might be bound by protein, and we wouldn't have nearly so much transcription going on. That's just one example. There are many scenarios you can imagine how multiple elements might affect when transcription occurs and how abundantly transcription occurs. Okay. Any questions about that before I move forward? You're a quiet group today, and I don't have a song for you. I apologize. I do have a joke, though. Actually, I heard a dumb joke. Okay? So, there's this guy who owes this other guy some money. All right? And he, his name is John. Okay? And he's, this guy who's owed the money comes to John's house looking for the money. And knocks on the door, and John's wife comes to the door, and he says, "Is John here?" And the wife says, "No, he's out getting some cotton." Okay, if you've heard this, please don't give away the dumb punchline. Okay? He's out getting some cotton. Okay. So the guy goes away and comes back. The next day, he comes up and knocks on the door. "Is John here?" "No, he's out picking some cotton, getting some cotton." This goes on for several days. Every day, same story. But the guy's really starting to get suspicious. You know, John doesn't want to pay me this money. I don't think he's out doing this. One day, comes to the door. "John here?" "No, John died." "John didn't die. John doesn't want to pay me my money." "He died." "No, he didn't." "Yes, he did." "No, he didn't." Argue. Go out and said, "Well, go out and look at his tombstone." The guy says, "Well, he's got a tombstone. He must be dead, right?" Goes out and looks at the tombstone. You know what the tombstone says? It says, "John, gone, but not for cotton." I've got a minute. I have another one, okay? I got more of a laugh than I thought. You guys are only encouraging me here, okay? This about the world's greatest piano tuner. Have you ever heard the world's greatest piano tuner joke? You have? You feel like it. Oh. Did I tell this last term?
I don't think so.
I don't think so. I don't think I told this in a long time. Okay, so this lady goes to this garage sale, right? And she's at the garage sale, and there's this beat-up old piano at the garage sale, and it says, you know, $50. She goes tink, tink, tink on it. It sounds horrible. And the lady at the garage sale says, "Well," she says, "this is a great piano. You just have to get it tuned up." She says, "I've got the greatest piano tuner in the world." "Yeah, sure." She says, "His name's Fred Abernathy. You have to look him up in the phone book and call him up. He'll tune your piano, and he does it really cheaply." So the lady figures, you know, $50, what am I out? So she gets this dilapidated old piano taken back to her house. She looks in the phone book, and sure enough, there's Fred Abernathy in the phone book. She calls him up, says, "I just bought a piano, and I need to get it tuned." He says, "No problem." He says, "I'll be right over, and I'll fix that piano for you." He comes over, and he tunes his piano, and what he does is amazing. The piano sounds like it's a grand piano, and she's got this thing for $50, plus whatever she paid him to tune the thing. This is incredible. She's just happy as she can be. You know, she's got this magnificent piano with this magnificent sound. She plays it, and she plays it, and she shows all of her friends, and she tells everybody about this. And about 6 months later, you know, the piano starts getting a little bit out of tune. "No problem. Call Fred." She calls Fred and says, "Hey, I need you to come tune my piano." And he said, "Didn't I tune your piano about 6 months ago?" And she says, "Well, yeah, you did. You did such a great job." He says, "I'm sorry." He says, "I can't do it again." She says, "Why not?" And he says, "Because Abernathy only tunes once." Abernathy only tunes. That one didn't get nearly as... It's usually reversed. Okay. Opportunity only. Yeah. Oh! Now we get it. I should have... Okay. That loosened things up a little bit. Eukaryotes are different from prokaryotes, but they do share some similarities in how they organize and do things. Eukaryotes organize their ribosomal RNA sequences on a common pre-RNA. It's called pre-RNA, and by the way, it was called a pre-RNA in the prokaryotic cells as well, because it's not the final mature RNA. Mature meaning the final product. Okay? In eukaryotic cells, there are the 3 bigger ribosomal RNAs. They're all located on the same long transcript. And these RNAs undergo some processing, meaning chemical modification and digestion to produce the final products. In ribosomal RNAs in eukaryotic cells, we see a lot more chemical modification of the ribosomal RNA sequences. That's what they're trying to show you here. These are various groups that are put onto those ribosomal RNAs. And after those modifications have occurred, we similarly see endonucleolytic cleavage like we saw in the prokaryotic cells to produce the final ribosomal RNAs. In eukaryotes, there are 4 ribosomal RNAs. 3 are handled by RNA polymerase I, and they're the 3 that you see right here. Okay. tRNAs are also made in made in bigger complexes that are processed down to smaller ones. tRNAs in eukaryotes, like in prokaryotes, are also chemically modified. You can see some of the modifications I'll point out here in a second. And we can see an unprocessed tRNA up here. The unprocessed tRNA has what's called a leader sequence, which is basically a 5' end that's got to be clipped off. And there's a nuclease that clips that guy off. There's also a nuclease that clips the far 3' end off. And then we see attachment, like we saw in prokaryotic cells, of a CCA at the 3' end. Okay? So these are processing events that are happening during the, manufacture, as it were, of the transfer RNAs in eukaryotes. Now, if you look at these sequences, you'll notice that these guys are made initially mostly as G's, C's, U's, and A's. There's a couple modified bases in there. But in this processing, we are actually converting some of these bases to very modified forms over here. For example, this guy here, pseudouridine. We don't see it anywhere except for in transfer RNAs. There's a T. Okay? Why does it have a T right there? Okay? As I said, there are things about the structure of tRNAs that we don't fully know. There's a D, okay, standing for, I just had it, dimethyladenosine, I think. Anyway, but the point is that quite a few modifications are happening. Here's a bunch of Ds here, okay? So the chemical modifications appear to be important for the tRNAs. It may be that these modifications actually tell the cell this is a tRNA. Okay? So there may be some specific modifications that tell the cell this is a tRNA, don't go chewing it up, because cells do tend to chew up their RNAs. Chewing up tRNAs would be a little counterproductive because there really wouldn't be much accomplished by chewing them up. Okay. Eukaryotic messenger RNAs are modified in several ways, and they're modified in several ways that we don't see at all in prokaryotes. Several ways. I'm probably not going to finish with this today, so I'll finish with it next time if I don't finish today. So there are 2 very noticeable modifications that happen to start. Okay? One is at the 5' end, there's something put onto the 5' end called a cap. Now the cap is a methylated form of guanine that is added after initiation has occurred. Initiation starts here, right here, and moves down. The cap is this region shown in red, and it is added after initiation has occurred. Now, it is a modified guanine that's up here. That's one thing. Actually has a methyl group that's critical. And it has a very unusual bond. This is the only place in nature we see a 5' to 5' bond. Why in the world put a 5' to 5' bond on there? Okay? Well, it turns out there's a very important reason that this happens. It turns out there's 2 important reasons. But the most obvious important reason is the fact that cells don't want to have messenger RNAs around all the time. They don't want to have them around all the time. A gene that's useful now might be something that's causing problems later. So cells, in addition to making RNA, make ribonucleases that break down RNA. Ribonucleases are everywhere. If you ever work in a lab that does RNA work, you'll discover that you've got to be absolutely paranoid about touching anything because you've got ribonucleases everywhere on your skin, anywhere that touches anything. And they're very, very hard to get rid of. They're there so they can break down RNAs when they're not needed. Okay? In prokaryotic cells, they're loaded with RNases as well. In prokaryotic cells, the lifetime of a messenger RNA is about 10 minutes. In eukaryotic cells, the lifetime of a messenger RNA is on the order of 24 hours. And it varies from one to another, but that's an average. One of the reasons that eukaryotic messenger RNAs are stabler is they don't get attacked at their 5' end by nucleases. This cap helps to protect the 5' end from nuclease degradation. That's kind of important. Okay? The reason it's important is the 5' end is where the most important information is. That's where translation is starting. We will see that messenger RNAs get degraded from their 3' end towards the 5' end. Okay? That means that even during the degradation process, there can still be some translation occurring. Degradation is not real rapid. Okay, so this cap provides one function, which is to protect against nucleolytic degradation. A second function we'll talk about later, but it plays a role in translation as well. So the cap plays a role in translation. That one's not obvious from what I've just said. A second thing that happens to eukaryotic messenger RNAs that's interesting and odd is that at the 3' end of eukaryotic messenger RNAs, this only happens to messenger RNAs, at the 3' end of eukaryotic messenger RNAs, a sequence called a poly A is added. Poly A meaning many A's. Now, the way in which the sequence is added is interesting. The RNA polymerase goes along and it makes its business, and it gets near the end to a sequence that's AAUAAA. This tells the RNA polymerase we're nearing the end, and it's almost time to put on the poly A. A cut is made. Okay, so a cut is made here. So the RNA polymerase has made a longer sequence, but a cut is made here, and a specific poly A polymerase comes in and puts on up to a few hundred A's on the end. Now, that poly A sequence is on there also as a way of determining how long this messenger RNA is going to be around. I told you that degradation starts from the 3' end, and it doesn't go very fast. The longer that this A is, the longer this sequence is going to be around. The shorter that this A is, the less time this sequence will be around. So poly A plays a role in determining the lifetime of a messenger RNA in eukaryotic cells. That's a good place to stop. Let's stop there, and I'll see you guys on Friday. How's it going?